Hi everyone, and welcome to a brand new episode of Omics in Action. I'm Amos, and I'll be kicking things off by introducing a topic that's not only fascinating, but absolutely essential to the future of agriculture: functional genomics and proteomics. And before you tune out thinking this sounds way too technical, don't worry, we are breaking it down in a way that's easy to understand and, more importantly, really exciting. With me today are my teammates Awangko, Haisley and Kenny, and together we will be walking you through how these technologies are helping us grow better, stronger and smarter crops, the kind we need to face today's climate challenges. So, why should we care? Let's take a step back for a second. Climate change isn't just about rising sea levels or polar bears losing ice. It's hitting our food system hard. Crops are dealing with more intense drought, unpredictable floods, heat waves and pests that didn't used to be a problem. That's a lot for a plant to handle and a lot for farmers to manage. The bottom line, if our crops suffer, our food supply suffers too. Now, we have made big strides with traditional plant breeding and we are still using it. But let's be honest, it can be slow and limited in precision. Breeding a new crop variety the old-fashioned way can take years, and even then, it's hard to know exactly which traits you are selecting for. It's a little like trying to solve a puzzle without knowing what the finished pictures looks like. That's where omic technologies come in. When we say omics, we are talking about powerful tools that let us look deep inside the biology of plants, at their genes, RNA, proteins and metabolites. To understand how they function, how they respond to stress, and how we can use that knowledge to improve them. Let's start with functional genomics. This focuses on the role of genes, what they do, when they turn on or off, and how their activity changes when a plant is stressed. For example, we can ask, which gene helps a plant survive drought, or what gene helps it absorb nutrients from poor soil. By identifying these key genes, scientists can edit or select for them, giving us crops that are better suited for challenging environments. But we can stop at genes. That's where proteomics come in. While genes are the instruction, proteins are the workers. They carry out the actual tasks inside the cell, like managing water, defending against pathogens, or repairing damage. Proteomics allows us to see which protein increase under stress, how they change after being made, and how they interact with one another. Think of it like this. If the genome is the blueprint of a building, then proteomics is the activity happening the inside. The lights turning on, the doors opening, the plumbing working. You need both to understand what's really going on. By using functional genomics and proteomics together, we are able to build a full picture of how plants operate, from the genetic level all the way to the real-world function. This helps scientists develop crops that aren't just higher yielding, but resilient, meaning they can handle extreme weather, use fewer chemicals, and still produce food. And that's exactly what we need as we face global challenges like climate change and population growth. So, that's the big picture. Up next, Awangko will take you through some real-life example of how functional genomics is already being used to improve crops like rice, wheat, and maize, and the impact these breakthroughs are having in the field.
Thank you, Amos, for that great introduction. Now, let's dive deeper into the fascinating world of functional genomics and explore how this cutting-edge field is transforming agriculture by enhancing crop resilience. Okay, I will share some compelling real-world examples to illustrate the impact of these investments. Okay, let's start it. One of the most revolutionary tools in functional genomics is CRISPR-Cas, which is a highly precise gene editing technology that's reshaped how we approach crop improvement. Also than that, CRISPRs allow scientists to target and modify specific genes with remarkable accuracy. For instance, in rice, researchers have used CRISPR to knock out genes such as OSCNG-C14 and OSCNG-C16. This experiment revealed that these genes play a critical role in heat tolerance, particularly through their involvement in calcium signaling pathways. Calcium signaling is a vital mechanism that plants use to respond to environmental stresses, such as extreme temperatures, which help them adapt and survive under challenging conditions. Okay, then let's start it with in wheat, another staple crop, which are among one of them are functional genomics, which has provided valuable insight into how plants cope with heat stresses. Researchers have conducted studies to identify which genes are activated when wheat is exposed to high temperatures. They discovered that genes like TAHSFA6E, ascorbate peroxidase, and beta amylase are significantly upregulated during heat stress. This gene helps stabilize cellular process and protect grain quality, ensuring that wheat can maintain its nutritional value and yield even when temperature soars. This kind of research is critical for developing heat-tolerant wheat varieties that can thrive in warming climates. Okay, let's talk beyond that. Beyond heat stress, functional genomics is also making strides in addressing other environmental challenges, such as drought and soil salinity, which are major threats to global agriculture. By editing genes like OSDST and OSRR22 in rice and ZMRRGOS8 in maize, scientists have achieved remarkable improvement in how these crops withstand water scarcity and high salt level. This genetic modification enables plants to better regulate water use and maintain growth under adverse conditions, offering a lifeline to farmers in regions prone to drought or saline soils. So, what does all mean in practical terms for farmer and society? Stronger, more stress-tolerant crops translate to fewer crop failures, higher and more stable yields, and a reduced need for chemical inputs like pesticide or excessive irrigation. This not only boosts farmers' livelihoods by improving productivity and reducing costs, but also has a far-reaching benefits for the environment. By relying less on water and chemicals, this crop contribute to more sustainable farming practice and help address persisting global challenges like food security in the face of climate changes. Moreover, functional genomics is revolutionizing the way we breed crops. Traditional breeding methods can be slow and unpredictable, often requiring years of trial and error to achieve desired traits. With functional genomics, scientists now can pinpoint the exact genes that are responsible for critical traits, such as stress tolerance or disease resistance, and incorporate them into new crop varieties much faster. This precision accelerates the development of improved crops, allowing us to stay ahead of evolving environmental challenges and meet the growing demand for food. Okay, next, I'm really excited to pass to one of my friends, which are Haisley, who will dive into the complementary field of proteomics. He will explain how studying protein gives us a deeper understanding of plant biology at the molecular level and why this perspective is just as crucial for advancing agriculture. That's all from me. Let's continue with the next person.
Thanks, Awangko.
So far, we've talked about genes and how they help us understand what a plant can do. But what about what the plant is actually doing right now, especially under stress? That's where proteomics comes in. Now, proteomics might sound like a complicated word, but it simply means the study of proteins, and proteins are the real workers inside a plant. They carry out nearly every function, from helping plants grow, to defending against disease, to responding to heat or drought. Let me give you a real-world example. Imagine a maize plant, or commonly known as corn, growing during a dry season. Its survival depends on how well it can respond to drought stress. Scientists look at two types of maize. One is drought tolerant and one is sensitive. They discovered that the drought tolerant maize made more of certain proteins, like aquaporins. These proteins help move water efficiently inside the plant, which is a critical ability during water shortage. Another group of proteins that often show up under stress are called heat shock proteins, or HSP for short. Despite the name, they don't just show up when it's hot. Their job is to protect other proteins from getting damaged or clumping together when the plant is under stress. Think of HSP like plant doctors. They help repair or stabilize the system when things go wrong. In wheat, for instance, researchers found that these proteins increased up to 5 times more when the plant was exposed to high temperatures. And there's more. Plants also go through quick chemical changes, known as post-translational modifications, where proteins are modified after they are made. One of the most common changes is phosphorylation, which is like flipping a switch to turn a protein on or off. This allows the plant to react quickly to its environment, for example, when it suddenly gets too hot or dry. So how exactly do scientists study all of this? Well, they use something called an advanced tool like mass spectrometry, which lets them identify and measure thousands of proteins at once. Oftentimes, they take the sample from the leaves because this is where the most visible stress response happen. Then, they compare stress plants with healthy ones to see which proteins change. Now, why does this matter for everyday life? Well, number one, proteomics can help improve food quality. By studying proteins change during crop development, scientists can enhance the nutritional value, texture, or shelf life of food. For instance, identifying proteins related to taste, allergen content, or spoilage means better and safer food products for the consumer. And number two, proteomics can help ensure food safety. Let's say we want to check if rice is contaminated or if it's developed harmful proteins due to disease or environmental conditions. With proteomic tools, we can detect that before it reaches your plate. But the real magic happens when proteomics is combined with field research. In one study, researchers exposed maize to drought multiple times and looked at the changes in protein levels. What they found was not only in water-related proteins, but also in those involved in photosynthesis, energy storage, and even DNA repair. These detailed insights help us figure out exactly which traits we should breed for if we want stronger, more reliable crops. In the long run, proteomics supports something called precision agriculture. This is where farmers can use real-time data like protein biomarkers to check their plant's health and take action early. For example, if a plant shows signs of early heat stress, the farmer can irrigate sooner or apply specific treatments. This means fewer crop losses, less waste, and more efficient farming. So to summarize, proteomics helps us move beyond just knowing what genes a plant has. It shows us how plants actually survive and adapt in real time. It gives us knowledge to, number one, build crops that can handle climate stress, and number two, improve food safety and quality, and number three, make smarter, data-driven farming decisions. In short, It helps us create crops that are not only productive but also resilient, nutritious, and safe. Now, I'll pass it over to Kenny, who'll wrap things up by exploring where all this science is heading next, including new tools and technologies for the future.
Thank you, Haisley. Now we have seen how the functional genomics and proteomics play its role. Let's look ahead, because the future of agriculture is all about putting this piece together. We are now in an era where scientists do not just look at the genes or proteins in isolation. They are actually combine data from genomics, transcriptomics, proteomics, and also metabolomics to get a full picture of how plants respond to the stress. This is known as a multi-omics approach, and it's helping us understand the plant system as a whole, rather than just one layer at a time. Take maize as an example. In a previous study, scientists used multi-omics to see how drugs affect everything. This including genes expression, protein activity, and also metabolite. Then using machine learning, they predict how well the plants would yield, with around 85% accuracy. This kind of insights can help farmers and breeders make smarter and faster decisions, way before a seed even goes into the soil. Then, there are also growing interest into epigenomics, which look at the change in genes expression that do not involve any altering the actual DNA. Things like DNA methylations help plants to remember the stress they met before, sort of like training their internal response system to react the stress faster next time. It's giving us a new way to build long-term resilience into crops. And there is a good news. These tools are become more accessible. This is because the cost of sequencing and proteomics analysis is dropping, which enable researchers and breeders in more parts of the world to use them. That means local crops in local climates can be improved using cutting-edge science. Then looking ahead, AI is also going to be a big part of this. With the help of AI and large omics dataset, scientists could soon predict plant traits like drought tolerance or nutrient use with more than 90% accuracy. Imagine being able to design a crop that's climate ready before it's even planted. That's where we are heading. And it's not just about higher yields. We are also talk about more nutritious crops that use water more efficiently and the needs fewer chemical inputs. This is about making agriculture smarter and more sustainable and future-proof. So, to wrap it up, functional genomics and proteomics are not just true in a lab. They are a part of growing toolkits that's helping us adapt to climate change, improve food security, and build a better food system from the ground out. Then, that's all from us. Thank you so much for tuning into Omics in Action. On behalf of Amos, Awangko, Haisley, and me, we hope you have enjoyed this journey into the world of modern plant science. And hey, the next time when you eat a bowl of rice or bite into a juicy cup of corns, just remember, this is a good chance omics has something to do with it. See you next time, and until then, stay curious, stay going.